Endocytosis is a fascinating cellular process that allows cells to bring large materials inside. Think of it as the cell's way of importing substances that are too big to simply pass through the membrane. This is a crucial mechanism that cells use constantly to survive and function properly. Throughout this video, we'll explore how this process works and why it's so important for life. So what exactly is endocytosis? It's a process where cells engulf external materials by forming membrane-bound vesicles that bring substances into the cell. Imagine the cell membrane wrapping around a substance like a bubble, pinching off to form a small package inside the cell. These packages are called vesicles. This mechanism allows cells to take in materials that cannot cross the membrane through simple diffusion or other transport methods. Why do cells need endocytosis? First, large molecules cannot cross the membrane directly. The cell membrane is selective, and many important substances are simply too large to pass through. Second, endocytosis allows uptake of nutrients, hormones, and other essential materials that cells need to survive. Finally, this process is essential for cell communication and regulation, helping cells respond to their environment and maintain proper function. There are three main types of endocytosis, each serving a different purpose. The first is Pinocytosis, the second is Phagocytosis, and the third is Receptor-Mediated Endocytosis. Each type has unique characteristics and is used by cells in different situations. Let's explore each one in detail to understand how they differ and when cells use them. Pinocytosis literally means cell drinking. This type of endocytosis involves the uptake of fluids and small dissolved particles. The cell membrane forms tiny pockets that fold inward, creating small vesicles filled with extracellular fluid. These vesicles bring in whatever dissolved substances happen to be in that fluid. It's a non-selective process, meaning the cell takes in whatever is present in the surrounding liquid. Phagocytosis means cell eating. This type is used for the uptake of large solid particles or microorganisms. The cell extends projections called pseudopods, which are temporary arm-like structures, to surround and engulf the material. Once surrounded, the material is pulled inside in a large vesicle. White blood cells use phagocytosis to engulf and destroy bacteria and other pathogens, making it crucial for our immune system. Receptor-mediated endocytosis is highly selective uptake of specific molecules. Special receptor, proteins on the cell surface bind to target molecules like a lock and key. When the right molecule binds to its receptor, it triggers the formation of a vesicle that brings that specific substance inside. A common example is cholesterol uptake via LDL receptors. This precision allows cells to take in exactly what they need. Let's recap the key points. Endocytosis brings materials into cells via vesicles, which are small membrane-bound packages. There are three types: pinocytosis for fluids, phagocytosis for large particles, and receptor-mediated for specific molecules. This process is essential for nutrient uptake and proper cell function. Finally, remember that endocytosis requires energy, making it an active transport process. Understanding endocytosis helps us appreciate how cells interact with their environment and maintain life.
If you like this video, hit that like button and don't forget to subscribe. Visit codelucky.com for more such useful content.